so much for the nice introduction. And Duangjai, and I'm sorry, but I just don't know how to pronounce your last name. So it's just I have to call you Duangjai, and I hope it's okay. But anyways, it's my great pleasure and honor to have this opportunity to talk about my recent researches in my lab. And before I start my talk, I'd like to thank the organizer and again, especially Duangjai, for inviting me over this great webinar series. And as the title indicates of this slide, which says application of deep eutectic solvents for efficient preparation of biopolymers and nanoparticles from natural resources. My presentation will be about this application of DESs and also especially, I guess, we can just call these deep eutectic solvents as natural deep eutectic solvents. But before I talk about my own research, maybe I can give you, the audience, a very brief introduction of where I am from. So I work at Sungkyunkwan University School of Pharmacy in South Korea, which is shown in this small circle. And currently, our School of Pharmacy is has a lot of partnerships or brother sisterhood with many prestigious schools, and we are eager to find any active collaborations. So after this talk and then later, just feel free to talk to me if anybody is interested in talking about future research collaborations. And anyways, like I told you in the first slide of this talk, my presentation will be covering the application of DESs or NADSs to efficiently prepare two different types of materials from two different natural resources. And the first part will be about the chitin-glucan complexes, which are bio-based polymers, and the resource will be mushroom. And the second part will be the biosynthesis of nanoparticles, especially silver nanoparticles, using green tea leaves to provide efficient phytochemicals. So I'd like to go on to the first part, which is about the chitin preparation using DESs. So there has been a lot of attention on biopolymers because biopolymers are sustainable and renewable, as everybody knows, and especially chitin is the second most abundant natural polymer after cellulose. And it's composed of N-acetyl-D-glucosamine units, as shown in here, and they're linked through this beta-1,4 linkage. And so basically, chitin is polysaccharide-based natural biopolymer, and it's quite attaining a lot of interest all over different fields because this chitin is biocompatible and biodegradable. So there are many papers to prepare this chitin from various sources and also like modify this chitin for better high value, like very useful materials. And chitin is easily found in insects, crustacean shells, fungi, and algae. And there are several methods, like several different kinds of methods to prepare chitin from natural resources. And as you can see from this slide, we can say that crustacean shells have been the most popular resources to prepare chitin. And when crustacean shells are used as the natural resources for chitin preparation, mostly chemical and biological methods have been used as the most like a common method. But recently, people also started to look to use ionic liquids for chitin preparation from crustacean shells. And also not many, although there are not many, but DESs have been started to used as chitin solvent for chitin preparation from crustacean shells. And very recently, a small number of studies have started to use mushroom for chitin preparation, but in this case, chemical method was applied. So what's wrong with these conventional or previously reported methods? I can say there are two aspects that we need to pay attention to. So first one is regarding the resources, the type of resources. As I told you, like I briefly introduced, crustacean shells are the most common natural resources for chitin preparation because they are just like a waste product and then because people just don't eat these shells. But there are some essential, potentially essential issue with the potential presence of allergenic protein in these crustacean shells. And then maybe another issue could be the seasonal variation in acquisition of these crustacean shells. And in comparison with these crustacean shells, mushrooms have quite advantages because mushrooms do not have any potentially allergenic proteins at all. So we don't need to worry about the complete removal of allergenic proteins as people do in crustacean shells. And cultivation of mushrooms is not affected by any seasonal variation. And another important aspect about mushroom is that there is no need for demineralization. In crustacean shells, there are a lot of minerals that need to be removed when they are prepared for chitin, but mushrooms do not need any that kind of step because there are not many minerals from the beginning. And the product, chitin product that is produced from mushroom is rather chitin-glucan complex than just a simple chitin. So mushrooms contain glucan in addition to chitin, and as a treat, the product of chitin products are more like a chitin-glucan complex in mushrooms. So from now on, I may refer this into just CGC for abbreviation. And another issue with the conventional or previous methods is, especially with the chemical methods, which are the most common methods till now, is that harsh conditions may be involved, especially like a very strong acidic conditions are involved in this chemical method, and therefore a lot of production of a lot of wastewater is unavoidable. And maybe biological methods are quite eco-friendly and safe, but still these kind of methods may have some problems such as high cost and maybe low efficiency. So in this study, we were thinking maybe DES can be applied to prepare chitin from mushrooms. And hopefully the method will be eco-friendly and very highly efficient. So this is what we did for this study. This shows the schematic for the mushroom that we used. So basically, we started with white button mushroom that was dried. And as you can see, beta-glucan and chitin is forming a complex, and then there is a protein outside or inside, whatever. Anyways, and this protein needs to be removed for chitin-glucan complex preparation. So conventional as a reference or comparative method, we applied sodium hydroxide, which is this chemical method, basically. So the pretreatment of this white button mushroom leads to the product called alkali insoluble material, or AIM. So we compared this AIM with our own products. For our method, we applied 5 different kinds of natural deep eutectic solvents, as shown in here. And what was interesting about this DS treatment for CGC preparation was that we could produce two different types of CGCs different from the conventional method. What I mean is that pellet can be directly prepared as this AIM was produced. So after treatment with DS of this mushroom, and then we can get pellet, which can be designated as DS_P. And at the same time, we ended up having supernatant after removal of this pellet. And we found that if we add a lot of water to this supernatant, we can also precipitate another type of CGC, which was designated as DS_S. S stands for supernatant and P for pellet. So, and all these three types of CGCs were further characterized, including protein content determination and chitin and glucan content, elemental analysis, FTIR, X-ray diffraction, thermogravimetric analysis, scanning electron microscopy, etc. So these are some examples of characterization results. And I briefly already told you that each DS, one type of DS could produce two types of CGCs, like _S and _P. So that was one interesting fact about our experimental results. And we found that all the DSs could produce two different types of CGCs, and all the CGCs were found to contain different compositions of chitin and glucan. And among the 5 different types of DSs, we kind of decided to focus on DS called BU, which comprises betaine and urea at certain molar ratio. And then because the reason for choosing BU for further investigation was because BU could remove protein the most, very similar to AI, like a sodium hydroxide based AIM. So basically protein removal, the more protein removal is more favorable for CGC. So we found that two types of CGCs from BU treatment had a quite different degrees of acetylation. If you remember the monomers of chitin, it has like acetyl group. And then when the acetylation degree is high, it's called rather chitin. And when it is more like deacetylated, and then it's usually referred to chitosan. Anyways, but what was more interesting about our results was that many like DES-based CGCs were found to have a very low crystallinity compared to the reference method or chitin, which is commercially available and was derived from crustacean shells. And even like, you know, raw mushroom has quite high crystallinity index. So it was quite interesting to see that DS could remarkably lower the crystallinity of CGC. And we found, despite the very similar different degrees of acetylation or chitin-glucan content, we found that those two types of CGCs showed very similar thermal properties to each other. And these CGCs also showed very similar thermal properties to that of AIM. And as we can see from these SEM images, this is the raw mushroom, like no treatment at all, and AIM, which was treated with sodium hydroxide as the conventional method. And these two are the CGCs that were pre-treated with BU, so as a pellet or supernatant. What was interesting was that the morphology of these two were very different from AIM. What I mean by very different was that the structures was very porous in both cases, and then the surface was quite rough compared to the smooth AIM. So from this study, we kind of concluded that DSs probably reduce hydrogen bonding and lowered crystallinity, which results in porous structure of these two CGCs. So in conclusion from this first part, we presented a new green method to prepare various CGCs from mushrooms. And we found that numerous CGCs that are free from potential allergenic proteins could be produced by applying various DESs. And the presented method is simple and involves only mild and eco-friendly conditions thanks to the use of DESs. And lastly, we suggest that mushrooms having no or almost low commercial values could be utilized as useful resources when we apply the NADSs to prepare chitin-glucan complexes. So that was the first part, and then I'll move on to the second part, which is about the biosynthesis of silver nanoparticles using green tea extracts that are produced by DESs. So metallic nanoparticles and then their modified versions are so popular and then extensively studied for variously wide applications from medicine to catalysts, cosmetics, and biosensors, etc. And of course, the silver nanoparticles are the most common, popularly studied nanoparticles among many different types of metallic nanoparticles. And silver nanoparticles are often applied in health, food, agriculture, or environmental applications. And there are generally two different approaches to biosynthesize silver nanoparticles, or actually any kind of metallic nanoparticles. And those approaches are top to bottom and bottom to top. And then these two approaches can include chemical methods or physical or even biological methods. But these conventional methods have some problems, such as toxic chemicals may be needed for chemical methods, and high energy consumption or surface imperfection may occur in physical methods, and maybe high costs will be needed. And biological methods are quite favorable because they are eco-friendly and quite safe, but there are still some issues with the potential contamination from microbes or other biological cells. So the microbes may be used in these biological methods. So many people believe that among numerous approaches in biological methods, probably plant extract-based biogenic synthesis of nanoparticles is the most desirable. In this type of synthesis methods, we do not need to add any other chemicals, but all we need is just the plant extracts that contain phytochemicals. And these phytochemicals can serve as natural reducing and end-capping agents at the same time. And the operation conditions are quite simple and then very safe. And what was better with this plant extract-based biogenic synthesized nanoparticles was that the nanoparticles are biocompatible thanks to the phytochemicals, and then they are known to have lower toxicity than just simply chemically or physically prepared nanoparticles. So this is what we try to do in this second type of study. So here we used green tea leaves as the natural resources to prepare green tea extracts. And to prepare green tea extracts, we used DESs And for the reference or comparison, we also used water to prepare different kinds of green tea extracts or GTEs from now on. And then we applied ultrasound assisted extraction and centrifugation, followed by appropriate dilution of these extracts for to produce green tea extracts, and then just a simple mixing of green tea extracts with silver nitrate aqueous solution led to the biosynthesis of silver nanoparticles after a certain period of time. And these synthesized silver nanoparticles were characterized using UV visible spectrophotometry or FTIR, dynamic light scattering, zeta potential, SAM, and EDX, XRD, and maybe more. And these are some examples of characterization results. These are the real images of the synthesis reaction solutions over time from 0 to 24 hours. And you can see the colors and then maybe the speed of color changing is different depending on the solvent or the solutions. And then from these results and also from this UV visible spectrum that shows the surface plasmon resonance peak that is indicative of silver nanoparticle synthesis, we found that DES-based GTEs could produce silver nanoparticles with varying efficiencies and varying properties. And what was more interesting was that some DES-based GTEs could synthesize small size silver nanoparticles at higher efficiency than just a simple water-based GTE. And among many other characterization results, I am showing you the energy dispersive X-ray results, which indicate the peaks of carbon and oxygen of silver nanoparticles. So from these results, we could conclude that phytochemicals and maybe DESs were observed onto the surface of silver nanoparticles. That means at least the DESs did not have any adverse effects on the synthesis. And then from this SAM analysis, we found that DESs were actually very helpful or useful to produce silver nanoparticles that are very small sized and then not narrow size distribution, which is very desirable properties for nanoparticles. And this X-ray diffraction diffractogram results suggested that DES-based GTEs were very effective for producing silver nanoparticles with single crystal phase. In contrast, the conventional method using just aqueous-based GTE could produce silver nanoparticles and also silver oxide nanoparticles, so not single phases. And just taking together all those characterization results and the chemical analysis results, we could propose a mechanism for the DES-GT-based biosynthesis of silver nanoparticles, which is shown or summarized in this figure. But because I'm running out of time, maybe just I can briefly explain these mechanisms in this summary slide. So in this study, we showed that DES-based green tea extracts could be used to synthesize silver nanoparticles for the first time. And we found that DESs can exert constructive effects on synthesis efficiency, particle size, and dispersion of nanoparticles. And we also are suggesting that DESs can serve as efficient extraction solvents for phytochemicals from green tea powders, and also they can serve as effective surface modifiers of silver nanoparticles themselves. So they have like a dual roles in this method. And lastly, we can suggest that green tea leaves that may have no commercial values could be still valuable resources in this DES-based silver nanoparticle biosynthesis method. with that, I'd like to stop my talk, and then I want to thank my lab members who and collaborators who contributed to this today's results. And lastly, I'd like to thank you for your kind attention.
Thank you very much for very attractive presentation from Professor Lee. So it is the time for question and discussion. So you can ask directly with the speaker or you can ask the question with the chat. But I would like to take this opportunity to ask the first question because your presentation is really eye-opening experience for me. I'm very, very impressed. So I have a question on your first part
Okay.
about the mushroom. So there are many mushroom species in Asia. So I would like to know which kind of mushroom are the major raw material for your research group currently. And did
That.
you
Oh yeah, I was just yes.
sorry, and I would like to know this you compare the result between the different species of interesting mushroom in Korea.
Right. That's a very important and interesting question. And honestly, definitely, I thought about this issue before, like when I was starting this project. And when I was discussing this theme with my student who just started to work on this, and she said, whenever she looked up papers about constituents and maybe this chitin preparation, only species that she saw was this white button mushroom. So we didn't have any other references. And then just to make sure everything works fine, we will just use this white button mushroom. But honestly, in Korea, white button mushroom is not so popular, or maybe it's probably something. I don't know the name, like academic name, but it's like a straw, very thin straw. And then that kind of mushroom is very cheap and also popular, but not this white button mushroom. So definitely I was thinking about testing other types which are much cheaper, and then but our defense for using this white button mushroom was that we just don't need to use high value or like merchandisable mushroom for this chitin glucan complex preparation. So if we can just get any kind of low or no commercial value mushroom, then it should be fine. But that's a very important issue.
Yeah, I really agree that you use the low potential mushroom for like not the valuable mushroom in the not the how say high economic value of the mushroom because in this way you can increase the value of the raw material. It's really clever and
Okay,
really
thank
nice. We have a question from the chat as well.
you.
So
I see a
is
question from the chatting room.
yes.
Right. So I see a question asking, is DSGT-based nanoparticles safe? Any form of testing for its safety? Yeah, that's a very important also and interesting question. And thank you, Emily. And honestly, we have not tested the safety itself for this DSGT-based nanoparticles, but like I kind of showed in the slide, the second part of my study is not yet has not been yet published, and then honestly, this study is under revision now. So hopefully it will be fine. So it's quite due, like the due date is coming very close. And anyways, so one of the reviewers' comments was about not the safety, but it was about the application issue. So as the application, we applied this DSGT-based nanoparticles to the cytotoxicity using cancer cell lines. And nanoparticles may have some toxicity issue, regardless of their like preparation methods. But people, experts in this field, claim that biosynthesized nanoparticles are like much more safe than just a chemically synthesized nanoparticles, because these nanoparticles are covered with phytochemicals that can have some kind of protective effect. And in our case, we didn't have a chance to compare the cytotoxicity of our own nanoparticles with just chemically synthesized nanoparticles. But obviously, cytotoxicity for a cancer cell line was observed. And I don't know if we can say it's not safe or it's safe, but that's a very fundamental issue that needs to be answered in the future.